Welcome everyone. Today we are going to learn about ion selectivity by ion channels. This topic is important because it had brought a Nobel Prize. At first, let me explain what is ion selectivity. Basically, all ion channels are very selective for a particular ion. Like potassium channels only allow potassiums, not sodium, calcium, magnesium, chloride or any other ions. Now, how this ion selectivity is achieved, we will learn in this video. I have written here all the important points that I will explain. In 1998, Roderick MacKinnon and his colleagues at Rockefeller University first provided the atomic resolution image of a bacterial potassium channel called KcsA. Now, the determination of the 3D structure of this bacterial potassium channel by X-ray crystallography revealed how exquisitely this ion selectivity is achieved. Now, when scientists compare the sequence and structure of other potassium channels, organisms from bacteria, fungi, and even humans, they observe that all share a common structure, and that is all are homotetramers. So all potassium channels are homotetramers. Now, tetra means 4 and homo means identical. That means potassium channels contain 4 identical subunits. Now, each subunit can be divided into 2 categories. Why I am mentioning the structure? Because to understand ion selectivity, the structure of ion channel should be learned at first. So each subunit can be categorized into 2. One is called 2Tm 1P and 6Tm 1P. Now, what is Tm? Tm means transmembrane alpha helix and P means 4 segment. Now, in case of 2Tm 1P, here 2 alpha helix is present and 1 4 segment is present in each subunit. Okay. And in case of 6Tm, there is 6 alpha helix, transmembrane alpha helix present and 1 P segment is present. Now, under this 2Tm 1P, the example of these bacterial potassium channels like KcsA present, and also one potassium channel is called potassium inward rectifier also present. And under the 6Tm 1P type of potassium channels, there is voltage gated potassium channels is present. Now, I will show here the structure of this KcsA in this video. As you can see, this is the structure of bacterial potassium channel KcsA. It contains 4 identical subunits, and each subunit contains 2 transmembrane alpha helix and a P segment. Now, in this P segment, there is a loop structure that actually makes the selectivity filter. So, loop structure shared by 4 subunits makes the real selectivity filter. Now, one thing I want to mention that this 4 segment is important for the selectivity, and specifically if you want to know what is the real selectivity filter, that is loop structure of this P segment is the real selectivity filter. Now, scientists have found that this P segment is important for the selectivity. Now, how they know this? Because when they compare the amino acid sequence of all other potassium channels, they found that these sequences are highly homologous in all potassium channels. Not only that, if you mutate certain amino acids in these potassium channels, then potassium channels cannot discriminate potassium ions over sodium ions. Also, if you replace the P segments of bacterial potassium channels with the mammalian potassium channels, that mammalian potassium channels can also transport potassium ions very easily and well. So that means this 4 segment has significant role in the selectivity. Now, this 2 point is very important, so I have demarcated with it red color, that is hydration cell and pentapeptide. Now, what is hydration cells? When ions are present inside of a cell, that means in the cytosol, or outside of a cell, that means in the extracellular fluid, they are surrounded by water molecules. That's why it is called hydration cells or cage. So, like sodium and potassium ions are surrounded by 8 water molecules. Okay. Now, water molecules contain oxygen atom, and that partial negative oxygen atoms try to neutralize or partially neutralize the positive ions like potassium or sodium. So they are surrounded by 8 water molecules that makes a cage-like structure, and inside that there is sodium or potassium ions are present. Now, here this pentapeptide is actually the main cause of this ion selectivity. So what I have mentioned here, that potassium channels contain 4 subunits. Each subunit contains 2 transmembrane alpha helix, a P segment, and that P segment contains a loop, and that in loop structure there is a pentapeptide sequence is present, that is signature pentapeptide sequence. So in all potassium channels will find this pentapeptide. This is very conserved, and I will write the pentapeptide here. That is glycine amino acids, tyrosine amino acid, glycine amino acid, X and threonine. Now, in case of KcsA, this X, that means any amino acids, is actually valine amino acids. Now, what happens when this potassium ions or sodium ions try to enter inside the channel? They have to strip off all the water molecules that surrounds during this hydration cells. So it has to strip off 8 water molecules to enter inside the channels. Now, this pentapeptide actually makes a similar or very geometrical structure that is very similar when this potassium ions is actually present in the hydration cells. What I want to mean that, as I mentioned, in the water molecule there is oxygen, and that interact with the potassium ions. 8 oxygens actually interact because 8 water molecules surround, and each water molecule contains 1 oxygen atom. So 8 water oxygen atoms actually interact or makes a bond. Now here in this pentapeptide, this peptide bond or backbone contains carbonyl oxygens, because we know the peptide bond is CONH bond. So each peptide bonds actually show 1 oxygen atom. Okay, so 4 oxygen atoms are showing by this backbone, and threonine also contains 1 side chain oxygen. So what happens here? 4 subunits. Each subunit contains this one p segments, that means 1 loop, and each loop contains this pentapeptide. So 4 subunits show this pentapeptide structure, and all this backbone and this threonine side chain contains oxygens. And in case of potassium channels, this oxygens makes a very similar geometry. So potassium ions that is present in the hydration cells can easily enter inside the cell, inside the channels, and can make bond with the oxygen atom. So it will not require, there is no requirement of energy, so there is no energy penalty. In this diagram, the pentapeptide has been shown, like glycine, tyrosine, glycine, valine and threonine, as I have mentioned, and all 4 subunits actually represent this pentapeptide, and they project the carbonyl oxygens, backbone carbonyl oxygens. As you can see here, 1, 2, 3, 4. These 4 carbonyl oxygens actually shown by 4 subunits that has the pentapeptide, and it makes a similar structure, similar geometry when the potassium ions is present in the hydration cells. Here also the pentapeptides, the backbone carbonyl oxygens, are actually 8 oxygens actually make this cage-like structure, so the potassium actually feels it the similar geometry, so it can easily enter inside the potassium channel. But here I want to mention here that, as you can see, the diameter of sodium is actually smaller than potassium. Now if you try to explain the site exclusions of this unselectivity, you cannot actually make any sense, because sodium is smaller and potassium is larger in diameter. So why not sodium can enter inside the selectivity filter, because it is smaller in size. Okay, but if you want to know, you will be surprised that potassium channels actually favor potassium ions over the sodium ions 1000 times. That means it prefers 1000 times potassium ions, not the sodium ions. Now how this is achieved? Because it is very phenomenal. Although this is a smaller in size, it cannot enter the potassium channels or selectivity filter. So basically what happens when sodium present in the hydration cell, it can interact with the 8 water molecules oxygens. But when it enters inside the potassium selectivity filter, the oxygen molecules that are present in this pentapeptide cannot interact with the potassium because it has a smaller diameter. Because it has a smaller diameter, all the oxygen atoms, I have only shown here 4 oxygens, but there is 8 what oxygens are actually projected in the selectivity filter to make a similar geometry, as it is present in the hydrogen cells. As I mentioned, they are all surrounded by 8 water molecules. So as because these sodium ions is smaller in diameter, so the oxygen atoms that are projected by this pentapeptide cannot fit. So it requires energy, so it will not enter, it will remain in the hydrogen cells. So that's why it is not actually can transport. So potassium channels cannot transport sodium. The same argument is true for calcium ions, because calcium also smaller in diameter, and as calcium contains 2 positive charges, so it makes tighter bond or a strong bond with oxygen water molecules than the sodium. So it will not actually, or potassium, it will not enter inside, and that actually the geometrical problem is present for calcium. Now, in 2011, the structure of sodium ion channel is also discovered, and how selectivity is achieved by these sodium ions, because sodium ion channels also allow sodium ions, not potassium, calcium, or any other ions. Okay, so they also actually determine how this selectivity is achieved. But there is a difference between these potassium ion channels and sodium ion channels, because in case of sodium ion channels, there are distinct type of sodium ion channels present in diverse species. They use different selectivity filter. As I have mentioned, in case of all potassium channels, this pentapeptide, signature pentapeptide, is very conserved. But in case of sodium channels, they use different selectivity filter. So that's the difference with sodium ion selectivity. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching, and always keep learning.